This video is all about DNA and how it copies itself. Do you remember when we talked about sexual reproduction and the life cycle of organisms? Well, remember, humans have 46 chromosomes in each of our cells. In a female's ovary, some of the cells go through meiosis to produce an egg cell. In a male's testes, some of the cells go through meiosis to produce sperm. Each of these gametes have half the number of chromosomes as the original cell. So each gamete has 23 chromosomes. Sometimes a sperm and egg comes together through fertilization to produce a zygote with 46 chromosomes. That zygote divides trillions of times by the process of mitosis to eventually produce a baby, then a full-grown adult. So why am I showing you this animation again? Well, what are those numbers inside of each one of those cells? What's this 46? What's this 23? They are chromosomes, or you can just call them DNA. Inside the nucleus of each of your cells, you have 46 long strands of DNA. When your cells get ready for cell division, either mitosis or meiosis, that DNA becomes really coiled. That is called a chromosome. So DNA and chromosomes are exactly the same thing. Chromosomes are just tightly coiled versions of the DNA. So what exactly is DNA? DNA is the genetic material inside all of our cells. Do you remember from a few months ago when we talked about the things that were common in all of the cells? We said cytoplasm, cell membrane, ribosomes, and DNA. So what is the point of genetic information? DNA is the instructions to make proteins, and proteins are responsible for doing millions of things in our body, like building cells, defending our body against viruses, creating all of our traits. This information, DNA, is passed down from your parents. Remember, your mom's egg has 23 chromosomes, combines with your dad's egg with 23 chromosomes to create you, with 46 chromosomes in each of your cells. So you got half of your DNA from your mom and half from your dad. When we solved genetic problems in Punnett squares, we talked a lot about genes. Genes, remember, are just segments of DNA that code for traits, like hair color or eye color or height. And to make our life easier, we just represented these genes or alleles as letters. In this example, dad's sperm carries the dominant H allele and mom's egg also carries the dominant H allele. During fertilization, the two H's come together to form a zygote with two big H's. We're just talking about segments of DNA which code for proteins that determine your traits. DNA is found in every single cell on Earth, from bacteria to amoebas to mushrooms to giraffes. Where is the DNA found? Well, in eukaryotic organisms, it is found in the nucleus, and in prokaryotic organisms, it's found in the cytoplasm. Remember, because prokaryotic organisms do not have a nucleus, therefore the DNA is in the cytoplasm. No. DNA is a type of nucleic acid, and it's made up of smaller parts called nucleotides. A nucleotide is also made up of smaller parts. Each DNA nucleotide has a phosphate group, a deoxyribose sugar, and a nitrogenous base. There are actually 4 different types of nitrogenous bases: guanine, adenine, cytosine, and thymine. Because those are long, crazy words, we typically just refer to them as the first letter, G, A, C, and T. These nucleotides come together in a variety of different orders to form DNA. The sugars and phosphates are always on the outside of the DNA, therefore creating a sugar-phosphate backbone that runs along the outside. Notice in this picture, sugar, phosphate, sugar, phosphate, sugar, phosphate, and all the way up. Notice on the other side, sugar, phosphate, sugar, phosphate, sugar, phosphate, and all the way up. So the outside is the sugar-phosphate backbone. The nitrogenous bases are always in the middle of the DNA molecule. The entire structure of DNA is shaped in a double helix. That means that there are 2 chains, hence the word double, and the 2 chains are twisted together. Remember, you have 46 of these really long double helix molecules in every single one of your cells. Now let's get a little more detailed with the structure of DNA. The nitrogenous bases, which are found on the inside of the DNA molecule, have special rules to follow. They can't just be put in any random position in the DNA. The rules are that the adenine on one side of the DNA always bonds to the thymine on the other side of the DNA. Therefore, A equals T. And a cytosine on one side of the DNA molecule always binds to a guanine on the other side of the DNA molecule. Therefore, C equals G. Look at the picture to the right. You'll see that whenever there's a G, it's next to a C. Whenever there is a T, it's next to an A, and vice versa. This rule is called the complementary base pairing rule. Here's a little summary animation. On the left, there are a bunch of nucleotides. See how each nucleotide is actually made of 3 parts: a phosphate group, a deoxyribose, and a nitrogenous base. Also notice that each base can be different. It could be A, C, T, or G. With the help of an enzyme, these nucleotides can come together to form DNA. This is only showing one side of the DNA molecule. Now we have to add the other side. If an A is the first nitrogenous base on the side of the molecule, then a T must go to the other side of the DNA. G with C, T with A with T, C with G. Let's try a practice problem. One strand of DNA has a nitrogenous base sequence AGTAC. What is the sequence of the complementary strand of DNA? Well, again, remember, A matches with T and C matches with G. So the sequence of the complementary strand on the other strand of the DNA must be TCA-TG. Look it, they match up perfectly. A with T, G with C, and vice versa. Notice something else about the DNA molecule. We have all the nitrogenous bases in the middle. And remember, there's a sugar phosphate backbone on the outside of this double helix molecule. Also, it's not in a helix because I couldn't figure out how to get this animation in a helix, but in real life, it would be all twisted up. Let's try another practice problem. In a double-stranded molecule of DNA, 20% of the nitrogenous bases are cytosine. What percentage are guanine? Okay, so we know that cytosine is equal to guanine, so if cytosine is 20%, that means guanine must also be 20%. Next question. What percentage are thymine? Okay, this is a little harder. If cytosine is 20% and guanine is also 20%, that means they total 40%. That leaves 60% left. Since thymine equals adenine, that must mean that thymine is 30% and adenine is 30%. How is that? Do you have questions on how that works? Write them down if you do. I know I've said this a few times, but remember, every single cell in your body has the exact same 46 chromosomes, or 46 strands of DNA. Well, why? We all started as a zygote, a product of fertilization. This zygote went through mitosis many, many times to eventually produce trillions of identical cells with identical DNA. Remember, mitosis is a process of making identical cells. So that should make sense. Every single one of our cells and DNA are identical because they were made by mitosis. Even though every single one of our cells are identical, we are all different from each other. What makes one person different from another? It is the difference in the order of their nitrogenous bases in their DNA. So my order of A's and T's and C's and G's are in a different order than yours. Because DNA is just the instructions to make proteins, which eventually make traits, if the instructions are different than your instructions, then we have different proteins and therefore different traits. For example, let's pretend person 1 has this sequence of bases, AATTCGACTACG. And let's pretend that sequence of DNA makes protein A, which is brown hair. Person 2 has a very similar DNA sequence, but it's slightly different. Notice there's a C and T in place of G and A. Because the DNA sequence or the instructions are slightly different, it makes a different protein. In this case, it makes protein B, which is blonde hair. So the reason we are all different from each other is because we have different sequences of genes or different sequences of DNA, which make different proteins, which give us different traits. In order to make every cell in your body have the exact same DNA, the DNA needs to copy itself. This is called DNA replication. Cells are constantly dividing, replicating, multiplying, whatever you want to call it. They are constantly dividing and dividing and dividing. In order for cells to divide, they need to pass through the cell cycle. G1, the cell grows. S, the DNA is copied. And G2, the cell grows some more. So what we are talking about is DNA replication, which happens in the S phase of interphase. Here's another picture showing you the same thing. During the S phase, the DNA or the chromosomes are copied. You start with one strand of DNA, and then after S phase, you have 2 strands. You probably actually memorized all this in the last unit, but I never explained what that meant. How was the DNA copied in the S phase of interphase? DNA replication is the process of copying DNA. This allows for every single cell in your body to have the exact same DNA. It is replicated right before the cell goes through mitosis. And remember, it occurs during S phase of interphase. So the point of DNA replication is simply to make more DNA. So how does DNA replication actually occur? Well, first the DNA strands break apart from each other. Then the new nucleotides are added to the strands according to the rules. Nitrogenous base A matches with nitrogenous base T. Nitrogenous base C matches with nitrogenous base G. Then they're all put together, and now we have 2 identical strands of DNA. Notice we started with a strand of DNA that goes ACGAT, and the other side goes TGCTA. After replication, we have 2 identical strands of DNA. They're identical to each other, and they're identical to the original strand of DNA. That's the point of DNA replication, to make more identical strands of DNA. Here's an animation to show you how it works. The double helix DNA first splits down the middle. Then new DNA nucleotides come in and match up with the old nucleotides following the base pairing rules. A's with T's and C's with G's. In the end, you'll notice we started with 1 strand of DNA and now we have 2 identical strands of DNA. When replication is complete, the cell is now ready to undergo mitosis or meiosis.